Let's keep the show moving here. We got a couple presentations left. Oh, there is our next speaker. He's already here, Dr. Howard Federoff. He's the CEO of ImmunoTherapeutics. How are you doing today, sir?
Great. How are you, Spencer?
I'm doing well. I'm doing well. You know, it's Wednesday, which means we're over the hump, coming down to the back half of the week. Take us home, Howard.
All right. It's my pleasure. It's great to
All
be
right.
with you. So today I'm going to be talking about an approach that probably is familiar, but let me start by recognizing our exceptional management team, many of whom have joined since the company went public in March of this year. A little timeline for overview. Through a reverse merger, we became public. We began and aggressively then secured the in-license to both mRNA and lipid nanoparticle patents. We raised about 55 million in capital. We focused on an IMSC company that we had already interacted with and acquired Novellus Therapeutics. And along the way, as I've just indicated, we recruited some exceptional people to the team, and we were uplisted onto NASDAQ just about 5 weeks ago. So the technology that I'll be speaking about that will undergird much of what Brooklyn ImmunoTherapeutics is going to be doing is focused on mRNA cell reprogramming, mRNA gene editing, NovaSlice, which is a trademarked gene editing protein, which I'll speak about extensively, and Tornado, which is an mRNA delivery lipid nanoparticle system. We see ourselves at the center of an emerging technology landscape that includes mRNA therapeutics, in vivo gene editing, non-viral genetic medicines, and iPSC-derived therapies. Leveraging all of these is part of the purview of Brooklyn ImmunoTherapeutics. If we consider for a moment the in-licensed mRNA-based approaches with the lipid nanoparticle technology, we can show, as seen on the left side, the use of these in combination so that we can make engineered cellular medicines. If we then deploy these in a slightly different way, we can encapsulate synthetic mRNA, including perhaps the mRNA encoding a gene editing protein, and then deliver that in vivo, being able to then tune the delivery to the particular organ and a particular cell type to develop in vivo gene editing medicines. The foundational mRNA cell reprogramming platform is highly efficient. It involves the introduction of 5 proteins that reprogram a somatic cell, as shown on the right, a fibroblast, into an induced pluripotent stem cell. It has low toxicity, it has a high percentage of cells that are transfected, and therefore the yield with regard to correctly reprogrammed cells lacking genomic alterations is quite substantial. Importantly, because of the nature of the way that the reprogramming is done, there is no chance for genome integration. The reprogramming can, quite interestingly, be combined with gene editing to streamline, as I'll describe later, the autologous therapies for genetic diseases. And this represents a portfolio of in-licensed patents, which are extensive. The unique gene editing approach involves this gene editing platform, and it is encoded by a synthetic RNA. It is rapid because of high expression. It leads to transient expression, which conveys specificity. It is amenable to non-viral delivery, such as the Tornado or LNP I described earlier. There is no risk of vector insertion, and it leverages in-licensed patents for the construction of this novel nuclease, which on the right side you can see it is, by virtue of its polypeptide, it has a clear IP landscape. It has high specificity, meaning it could recognize collinear DNA of a large number of base pairs from 36 to 40. And interestingly, it is blocked by histone modifications, meaning that the genomic context in which it can produce editing is focused on the target and does not lead to so-called off-target effects, in part owing to the specificity, but also as well to the histone context. And it offers unlimited genomic site selection because it does not require, as does a CRISPR derivative, a PAM sequence. It can be used for gene editing, for gene correction, as well as for safe harbor insertion. And I'll just talk a little bit about these very briefly. The novel lipid, which we spoke about, the tornado, this LNP, displays low toxicity after repeated cell culture exposure, transfection. It is an efficient transfection method, even in the context of serum, and it can transfect many cell types. As shown here, this is an induced pluripotent stem cell that was transfected using the LNP tornado, carrying a synthetic RNA that directs the expression of a fluorescent protein. But when combined with a LNP tornado, can be also used to introduce genes into the ventricles of the cerebroventricular system in the brain, the neuroretina, as well as into visceral organs, as diagrammed here, the liver. I'll talk a little bit about initially our allogeneic stem cell platform, the IMSC, and mention that it is being pursued for acute respiratory distress syndrome by a company we own an interest in, Novocyte, and it is being developed for bone marrow transplant failure, poor graft function, as well as when gene edited for a variety of tumor indications. The allogeneic IMSC leverages all that I've described to date, but importantly, because it derives from a single induced pluripotent stem cell, its origins are very clear, so it addresses the issue of identity, purity, and potency. By going this route, it produces a low risk of toxicity, as do other MSCs. It is, like other MSCs, portending low immunogenicity, therefore lacking the requirement for immunosuppressive drug administration. It leverages decades of work on how to do process development and manufacturing. And we envisage a single drug product that can be used across multiple and varied clinical indications. The iPSC itself can be gene edited, so it can then reprogram or program the IMSC with additional properties that will expand the clinical indication space. The MSC therapies have had inconsistent historical clinical efficacy owing largely to product heterogeneity, which I outline on this slide, where you can see that the historical issues due to donor variability, tissue source variability, manufacturing variability, limited or inconsistent characterization, poor mechanistic understanding, all of which are addressed by using the IMSC approach. And particularly because we have such an efficient way to do gene editing, it addresses the ability to then gene edit the iPSC, and as I described moments ago, then drive the differentiated iPSC into an IMSC that has new properties. The IMSC application in graft failure and poor graft function relates to the phenomenon that MSCs modulate the immunological responses, support hematopoiesis, and can repair bone marrow stroma that often are damaged in the setting of antecedent chemotherapy needed to condition patients. The clinical applications in hematopoietic stem cell transplant, HSCT, will allow us to approach treating engraftment failure or poor graft function following transplantation, but as well promoting HSC engraftment in the setting of de novo transplantation. Working with world-class key opinion leaders in HSCTs, we are now focusing on the best clinical applications and trial design and anticipate entering into the clinic first in human in Q4 of 2023. Developing a family of gene editing iMSCs borrows on that which we've just discussed, which is the unedited iPSC-derived iMSC, and allows us to address a variety of solid tumors where there is clinical high unmet medical need. The gene editing at the iPSC level, and then the thorough characterization of that edited product, followed by differentiation, is the path forward. We illustrate here that we can insert interleukin 7 and interleukin 15 genes into a safe harbor location that will then drive expansion and engraftment in the setting of a variety of hematological malignancies. And moreover, this approach can be combined with CAR-T and checkpoint inhibitors. This diagram just lays out the process as we currently envisage. In the autologous cell therapy domain, we believe that the efficient mRNA gene editing and the efficient nanolipid particle tornado will allow us to take CD34 positive cells and then to gene edit them, ultimately allowing for those cells, once edited and free of chromosomal abnormalities, to be re-administered to patients for hemoglobinopathies, primary immunodeficiencies, and congenital cytopenias. This leverages 3 decades of clinical experience isolating CD34 positive HSCs. And moreover, we know that these cells can robustly engraft and when administered back into patients have a tremendous safety record. The mRNA-based gene editing is less complex than other methods such as viral methods, and certainly is safer than those viral vectors that lead to proviral genome copy insertion. This offers substantial opportunity for future partnering. As we think about autologous iPSC therapies, the technology, again, that we've been speaking about is the safest, most efficient, and fastest method for iPSC derivation. It is safe because it is a non-integrating approach using synthetic RNA to synthesize the reprogramming of factors. It is efficient because it uses the lipid nanoparticle approach that we described, which can be then repeatedly applied to cells to lead to efficient reprogramming, and as I mentioned earlier, concurrent gene editing if warranted. And it is fast in that reprogramming and iPSC colony formation occurs within 2 weeks. The safety and reliability and speed enable these autologous iPSC programs, and this efficiency permits the reprogramming of low quantities of cells from biopsies and simultaneous correction of gene defects. We can quickly produce multiple iPSC clones per patient, and the absence of genome integration facilitates screening to identify and characterize a safe clone to move forward into further development. As an example, the autologous iPSC gene-modified autologous iPSC can be used for paroxysmal nocturnal hemoglobinuria, where a somatic mutation in the bone marrow, but not affecting other somatic tissues, does not require gene editing, and therefore it can be envisaged that this approach can be used to restore normal bone marrow function in the setting of carrying out iPSC-directed reconstitution. When we use gene editing, we can take on infectious diseases and other diseases which are of a monogenic variety. The cartoon here just basically illustrates our current understanding of how this will unfold when we move forward translationally and into humans. In vivo gene editing technologies are also leveraged using the nanolipid particle, the tornado approach. We have composition of matter IP. We can tune the nanolipid particle by varying the fatty acid length, the degree of desaturation, and the like. And we can deliver nucleic acids, either RNA and or DNA, which facilitates gene correction approaches. The proprietary site-specific nuclease, which I spoke about, NovaSlice, it can target any gene through design of the protein binding domains. It is highly specific to target the genomic sites and achieves high-level but transient expression, which enhances safety and likely portends lower immunogenicity. As we see this currently, the in vivo gene editing approach will involve, as I said, the nanolipid particle tornado with the synthetic RNA, which can then be used to transfect the choroid plexus, the neuroretina, or the liver to take on this monogenic disorder known as familial transthyretin amyloidosis, which actually produces clinical features in a variety of both central and peripheral targets, and therefore contemplating how to direct editing to each of those is clearly important from a clinical concept, as well as having the potential to have a single drug product delivered to the appropriate location, which is enabled by which mutations are likely needing to be corrected in which anatomical compartment. So if we consider this for a moment, this is a disorder that in the non-familial form involves patients up to a 500,000 worldwide. In the familial form, approximately 10-fold less. This will be our focus. Patients who present with symptoms typically in the peripheral neuropathy or cardiomyopathy live for 5 to 15 years unless some intervention is taken. It is autosomal dominant. It is owing to multiple mutations in the gene. The mutations increase the propensity of the encoded protein to organize into what's known as amyloid, and it is the amyloid deposition that is toxic. And the genotype-phenotype data suggests that we know where and in which location anatomically we need to do the gene editing in order to produce the appropriate clinical benefit, whether that be in the brain, the choroid plexus, the retina, or the liver. So the approach that we take is one that actually is amenable to all of the clinical features, the polyneuropathy, the cardiomyopathy, the retinopathy, and the cognitive deficits that are associated with certain mutations in each of these anatomical compartments. The Brooklyn approach can take on all of these. Our pipeline looks like this as illustrated. I mentioned acute respiratory distress syndrome and its prosecution by Novocyte. This is an unmodified or naive IMSC. Our BMT and HSC approach, our solid tumor approach with the gene edited form of the IMSC, and other oncological indications. The autologous HSC approach, we are now focusing on one of several candidates, and I just described at a high level some of the considerations in terms of clinical indications. And our autologous approach for the iPSC-derived cell therapy for paroxysmal nocturnal hemoglobinuria is the one that is most front and center. In the setting of in vivo gene editing, the transthyretin amyloidoses, as I described, which can affect the brain, the eye, and the body in terms of the heart and the peripheral nervous system, is a set of clinical indications that can, in our view, be addressed by a single gene product and be delivered to the correct anatomical compartment depending upon which mutation and which area is affected by the amyloidoses. So in summary, Brooklyn will leverage best-in-class mRNA-based technologies to deliver transformative regenerative medicine. We have exclusive license to foundational IP, mRNA, and gene editing. We have a diversified product strategy for multiple clinical indications. We have an experienced management team with a deep expertise in cell and gene therapy, strong partnership possibilities for translational execution and future innovation, and the in-licensed patent portfolio offers the possibility for sub-licensing opportunities. Thanks very much.
All right. Thank you, Howard Federoff, the CEO and president of Brooklyn ImmunoTherapeutics. Sorry, ticker is up on the screen there, BTX. If we have any questions for Howard, now would be the time to ask them. I will just ask you a question that's frankly I ask a lot of people. I've asked it several times today, which is, what exactly is the most difficult thing right now?
Well, I think if you generalize the question, I think many of us that are building companies want to be efficient in use of capital because it benefits shareholders and future investors. So thinking about the prioritization and thinking about the time to demonstrate in the clinic that there is biology and often clinical benefit, I think is the path forward.
Awesome. All right. Dr. Federoff, thank you so much for joining us today on the Benzinga Small Cap Conference.
Thank you, Spencer, and thank you to the audience.